Very good. So welcome to our main event. My name is Eric Novick. I'm your host. This is our inaugural virtual event. We're trying out this new platform, and we're trying out the new format. The format that we're using, it's sort of like if you've ever been to a movie screening where the director comes on before the show and we ask her a few questions about her background. Then we see the movie, and then the audience gets to ask a few questions. And so this is how we are going to run this. I hope you love it. If you don't, let us know. If you hate it, let us know. We can always modify it. So with that, I'd like to thank Jacqueline for spending the time with us. Welcome to the program, Jacqueline.
Hi, thank you.
All right. So just so that we get to know you a little bit better before we get going, I was looking at your background, and turns out as an undergrad you were a comp lit and biology major, and then I was kind of shocked to learn that you've read more Tolstoy than I had, and I was born in Russia and had to read that boring and verbose writer. So my question is not so much why you're interested in Tolstoy, but what attracted you to stats and to biostats?
Well, funny story about my papers on Tolstoy. I a lot of times would write, sort of summarize frequency of word counts in the book as part of the analysis. So I've always had a kind of quantitative approach to things. Grew up hearing a lot about market research analysis. My mom is a statistician as well, and was really motivated to apply that to the more important problems in biology. There's a rich family of approaches that we can take, and I saw that there was room to improve how we were doing analysis when I started learning about public health.
Interesting. And what was your first job after you graduated?
I took a job working at a clinical research organization in Boston, part of Harvard Medical School. And there we were running very large clinical trials, multi-center in cardiology. And at the time that I got there, we were managing data in Excel. I quickly tried to improve that process. Then got really interested in statistics and how we were analyzing the data for those trials.
All right. So now after that, I understand you kind of pivoted, and you were for a bit a director of data science at Penguin Random House. So selling books, bioinformatics must have tons of things in common, right? What are some of those things?
Yeah. Well, I did a brief stint working in genetics research for Alzheimer's disease, and there we did a lot of family cohort analysis. And so that's what got me really interested in multi-level modeling and hierarchical approaches. And it turns out that books published by an author behave very similarly to people in a family. And there's sort of a similar set of problems there around trying to estimate the relatedness of titles and sales curves.
Interesting. Okay. And so then you transitioned back to healthcare. So selling books must have not been that exciting. And you were eventually lead biostatistician at the Institute for Next Generation Healthcare at the Mount Sinai Icahn School of Medicine. So what were some of the problems that you worked on there?
So there we were working in mostly oncology, solid tumors, and immunotherapies. So there were two projects that I was involved in around biomarker discovery in clinical cohorts. So that's where a lot of the sort of modeling techniques that I'm going to talk about today were developed initially. Well, at least the approaches. I started playing around with them.
Okay. So as a kind of the precursor to the work you're about to discuss, I think a couple of years you wrote a paper with Sam Brillman and others titled Joint Longitudinal and Time to Event Models for Multi-Level Hierarchical Data. Now, for those viewers who are not from the healthcare or biostat space, the joint models here, have to do with a model for a time course of a disease, like a biomarker, and then a hazard model, and we estimate all that stuff jointly. But these models have been around for some time. I think I saw some references from the 70s on the structure of these models. What was kind of unique contribution in that particular paper?
So there we were trying to address a problem where there's sort of a family of biomarkers that are related within a patient. So the typical example is if you're doing an eye study and people have two eyes, and so you're tracking data for each eye, but the sort of combination of those two biomarkers, you want to take a shared parameter from the two of them and include that in the survival model. And the application that I was working on with Sam involved multiple lesions within the body, or multiple sites of tumor within the body, and sort of tracking, modeling the independent growth parameters, but then including the sum of those parameters in the survival model. And that lends itself to different extensions, taking the fastest growing lesion, taking the largest lesion, and estimating the impact of those on survival differently.
Gotcha. And was it also one of the first attempts to do a fully Bayesian analysis of these models? And then I believe you guys published a package an R package to use these and sort of with a formula interface, right?
Right. So there are a couple R packages that do joint modeling. A lot of them, like JM-Bayes, was using a Gibbs sampler. In this case, we sort of implemented these models using STAN. I know that there's some, I think Rosopoulos is also working on that as part of his package. The second component of our implementation is that it's fairly flexible and sort of has all the, can make use of the wonderful community of tools around STAN modeling, especially in R. And so I think it was one of the first to use full Bayes. I think I haven't looked at the actual timing of when we released it versus JM-Bayes.
Right. And of course, the follow-on work involved the large multi-trial models, and this is what we're going to discuss next. Mm-hmm. So with that, I'm going to turn it over to you. This next section, I think we have about 35 to 40 minutes planned. We're going to do a Q&A at approximately 12:45, followed by a post-presentation session in the sessions for those of you who want to stick around. So without further ado, I'll turn it over to you, Jackie.
Okay, thanks. So I'm going to talk today about predicting survival from early tumor data in oncology trials. So in the sort of application area of oncology. So a little background for those who are not familiar with the clinical trial development process. The sort of use case that we're prototyping here is what's called a go-no-go decision between phase 2 and phase 3. So I want you to sort of imagine that you're working on a new compound in either a biotech or pharma company. It's gone through the sort of preclinical and phase 1 testing. It's gone through phase 2, where we've enrolled sort of 30 patients or 40 patients. And now your decision is, or you're informing the decision of whether to advance it to phase 3. And this is a very critical decision for a number of reasons. First of all, the phase 3 trial is extraordinarily expensive relative to phase 2 trial. It can take a long time to run and enrolls a lot of patients. And so there's a huge opportunity and monetary cost to running a phase 3 trial. We can only take a certain number of them forward. And there are a lot more drugs in phase 2. So we want to be sort of careful in which drugs we select to take forward to phase 3. There are a lot of things that we're going to look at, or that you in that role would look at. First, the sort of quality of the drug itself, like what are the different aspects of manufacturing. That's not relevant in this case. We're not going to model that. There's the drug safety, and this we're also not going to model. And instead, we're going to focus on the drug efficacy, which is a big part It's a big part of that decision, but it's not the only factor. So we're going to look at a sort of typical phase 2b trial design. We're going to assume that we've enrolled around 30 patients in one or more treatment arms and that there's no control arm. And periodically, there are scheduled interim analyses, and then there's a final analysis. And at each of these time points, there is a decision that has to be made of whether to sort of continue on or stop the trial early or proceed to a phase 3 study. So our goal is to estimate the relative efficacy for this new compound X. When we say relative efficacy, we mean relative to existing treatments. In this case, we're going to be looking at non-small cell lung cancer. The complication is that we want to estimate this for the sort of expected or intended patient population. If we're looking at a drug that targets PD-1 or PD-L1, then we might look in a population of patients with high expression of that biomarker or presence seen on different assays. And so a complication is that we don't always know the population ahead of time. It may be that patients with certain characteristics are not good candidates for treatment, and we don't discover that ahead of time. Instead, we want to do this for each of the patient populations under consideration. And this would include pre-specified subgroups of patients, but also exploratory subgroups that are then considered after looking at the data. Again, this is going to be hard because we only have data from around 30 patients. And so we're trying to make a big decision with just a small amount of data. And so that's the sort of setup of what we're trying to accomplish here. We're going to start by simulating some data, right? This seems sort of futile, but to some, but to statisticians, this is how we gain some confidence in whether a method is going to work for the use case that we are interested in. I'll start by simulating data for existing therapies. So we're going to assume that there are 5 therapies and we have data for those 5 therapies in a number of prior studies. Each therapy is going to have a specific overall efficacy, and then some of the therapies are targeted, so the efficacy will vary by cohort. Some of the trials, not every trial includes every cohort, and then the efficacy is going to vary across studies just through random noise. And then we have sort of controls, but each study is going to have a slightly different control treatment. That treatment, the control treatment, is going to change over time. So we want to simulate a realistic scenario because when we go to analyze actual trial data, we find that there is a lot of, simply estimating the relative efficacy for existing treatments from completed trials is itself, a body of work and a hard problem to solve. So simulating realistic data will tell us how well we're doing in that effort. So I'm starting by simulating, sort of drawing randomly from a prior distribution for each of our treatments. I think in this particular run, I have 6 treatments and not 5. For this demonstration, we're going to have a fixed control, but in our sort of testing, we often will simulate several related controls. Here's an example of the simulated data from a single sort of replicate. And so given these treatment efficacies, we're then going to simulate patient data for 10 studies. And this is sort of the Kaplan-Meier estimate. And finally, we're going to simulate a set of data for our hypothetical phase 2 study. This is going to be for a new treatment. What we're going to do is simulate the full trial data. Then we're going to select 30 subjects at random to be in our treatment arm. And then we're going to censor the data early for each of the interim analysis time points. This is going to allow us to test whether or not we can recover the sort of full trial results from the subset of data that we're looking at. So at this point, we're going to fit our model. And I'm going to go into the details of the model later on in this talk, but I did want to at least give you a high-level view of how well the model performs and how closely does it address this use case. And here is the data for our phase 2 study. In the green, this is the limited survival data that we have available at this first interim analysis. And then these are the background studies. So in order to estimate the relative treatment efficacy, we want to include these background studies in the model. And we want to do this for two reasons. One is that it stabilizes our estimates. It gives us more realistic estimates for all the different components within this model. 30 patients is simply not enough to estimate these parameters reliably. And we have reason to believe that these studies will behave similarly, that there are aspects of patients that are shared across these studies. And the second reason is so that we can compare these efficacies to one another on the scale that we're interested in. So just to zoom in on the phase 2 study data result, you'll notice that this predicted survival is somewhat off from the observed Kaplan-Meier, the early survival data that we included. The reason for this is a little bit nuanced, but if we take our 30 patients up to our truncated time point, we can then extend that with the simulated data that we drew those 30 patients from. And this is what the KM curve would look like for those patients if we continued the follow-up longer. But then we also are interested in what would this curve look like if we didn't restrict it to the 30 patient subset, but instead we enrolled 300 patients and we also included a control arm. So this is the data that we're interested in predicting, is the eventual phase 3 trial population, not the 30 patient subset. And the reason for this is because the 30 patients may be, just by random chance, they may have larger or smaller tumors. There may be some imbalance in the selection for the populations for different key covariates. And when we want to estimate the overall efficacy, we need to adjust for that. And secondly, if we are planning the phase 3 trial population, we want to get a realistic estimate for that expectation. And so now we see that the observed Kaplan-Meier estimate for the true phase 3 trial population is at least consistent with the predicted survival over time. The sort of difficult part of this is that we don't have a lot of information for this trial about how the baseline hazard and how the population is different from these other studies. And so as we would expect to see a much wider range of uncertainty for these estimates than we see for other studies. And this is one of the things that we do in order to see evaluate how well the model is working. The second question you might ask is, how well can we estimate the treatment efficacy? This is something that we're very interested in recovering. The predicted survival includes sort of covariate specific prognostic effects plus a patient specific treatment efficacy estimate. And so the efficacy is arguably an easier problem than survival. And in this case, we do pretty well. It's not clear how well we're doing unless we repeat this many, many times. So that's the next step. We then run many, many draws of simulated data from the same parameters and evaluate how well we do in recovering both the efficacy and the survival estimate. And then I'm going to show you some examples from replicates that we've done. In some cases, they work really well. In other cases, they're a little bit off. So this is a set of data showing 2 replicates. We do a lot better with the background studies than with the new treatment. And a lot of this success depends on how we set up the simulation and how closely the simulation matches the model that we're fitting. So we want to test for robustness to model misspecification. So now I'm going to walk through sort of the mechanics of how this works. And this is going to be sort of both a high level for people who are more clinically focused, as well as I'm trying to get this balance between the clinical user and the statistical audience. the overview of the process is that I'm going to sort of walk through the model, given the tumor size data. We're then going to extract as much information as we can from the tumor size data that are observed over time. We're going to translate those signals to their survival impact. We're going to post-stratify to the phase 3 population or a different subpopulation of interest. And then we want to summarize our results on an interpretable scale. So the interpretable scale might be predicted median survival, or it might be the hazard ratio relative to a particular control treatment. But we ideally want to summarize our results on that scale so that it's very clear to decision makers what information is, sort of what the sum of information that impact is on the outcome, but also so that they can use their intuition in interpreting whether or not the result makes sense. You know, a hazard ratio of 2, you would look at that and say, oh, no, that can't be. Like, it seems implausible. So starting with the simulated tumor size trajectories, here we're looking at the sort of 30 patients in the early first interim analysis for our simulated phase 2 data. We're going to fit a tumor size model. We're going to sort of fit a model to this trajectory. And the model is going to have this form. It's a sum of 2 exponentials. F and 1 minus F represent the portions of cells that are either treatment responsive or treatment resistant. And then the cells that are treatment responsive shrink at this rate governed by Ks. And the cells that are resistant will grow by a rate governed by this Kg parameter.
Here are some examples of the types of trajectories that the model that Jacqueline was describing can support. On the upper right, we have lots of responding cells, but the resistant cells, the Kgs in the model, grow faster than the responding cells die. When that happens, we start relatively flat, but then we start seeing some growth. On the upper right, the situation is slightly different. There are lots of resistant cells, and those cells, the Kgs, grow faster than the responding cells die. This is the worst possible scenario, and this is where we see the fast progressors, immediate growth. On the lower left, we have lots of responding cells, which is good, and the resistant cells grow slower than the responding cells die. And this is, in general, the best of both worlds. And so we see an immediate response followed by stable disease. And in the lower right-hand corner, we have a situation that is approximately in between, where there are about half resistant and half responding cells. The resistant cells grow slower than responding cells die. So you see the initial response followed by the regrowth. And so these are just some of the trajectories that are possible under the model that Jacqueline is demonstrating.
Then what we're going to do is take these estimated parameters, we're going to estimate them for each patient, and then we're going to combine them to compute these 2 derived quantities that summarize the characteristics of the trajectory. So F and Ks are combined to produce a tumor shrinkage parameter for each patient, and 1 minus F and Kg are summarized to produce this tumor regrowth. And if we look at the distribution of these derived parameters across a set of real patients in non-small cell lung cancer, we'll see that there are a set of patients with low shrinkage and high regrowth. And these are in this long tail here. These are likely treatment resistant. They're likely aggressive growing tumors. We see most patients in the advanced metastatic disease are in this high shrinkage and high regrowth category. These patients are treated with either TKI or immunotherapy or chemo or various combinations of those. And so we do still see majority of patients are in this category of high shrinkage and high regrowth. And then in this particular data set, there's a set of patients that have high shrinkage and low regrowth. These are going to be the very strong responders to treatment and possible, long-term cure patients. And in this data set, they're mostly stage 3 with curative intent, sort of treated earlier in their disease history. The second feature of this is that among the patients who are in the stage 4 category, most of the variation occurs on the tumor shrinkage scale axis, that there's relatively less variability between patients on the tumor regrowth parameter. These parameters are nonetheless, they're rescaled in order to be on sort of comparable. So they have, these are the marginal distributions across all the patients, and they do have similar median values and similar variances. So going back to our example, if we pull out these are simulated patients, if we pull out 2 patients from our 30 patient subset and we fit the model to them, this is just the tumor size model, we are able to recover the parameters that for this patient reliably, and thus able to predict out of sample. So these open circles are the future observations that were excluded because we truncated the data early. And this is a separate patient in that 30 patient cohort where there is an estimate of more regrowth because of this final observation. And so we see this prediction sort of explodes out. So we're going to compute these per patient for each of our 30 patients. These are the 30 patients. And on this basis, we're going to try to estimate the survival of these patients, sort of predict their future survival. That we're going to bring these 2 parameters into our survival model. This is the structure of the model at a high level. It's a standard Cox proportional hazards model. However, it's also a meta-analytic model. Each of the parameters has a sort of shared variance across studies, is estimated per study, and then the effects are pooled across the studies. So we have the baseline hazard, which is stratified. All prognostic covariates would go into this term. In the context of the simulated data, we're not including strong prognostic covariates, although we do have simulations that do include them, but in this case, we're not using them. And then there's the so-called association. So this is where each of the tumor shrinkage and tumor regrowth parameters are going to inform survival according to this parameter alpha here. Finally, we want to summarize the results on an interpretable scale. Given the parameter estimates, we're going to generate posterior quantities of overall survival, median survival, treatment efficacy, in some cases, probability of technical success for particular trial design, particular sort of statistical analysis plan. Because we can simulate from this model, there's a whole range of things that we can do once we fit it to the data. We can predict ORR, or we can predict things under different circumstances. Details about this are that the tumor parameters, the F and the KS, and 1 minus F and KG, these get estimated, each one is a component GLM. F may be impacted by treatment, but it's also varies by gender, or it varies by histology. And in the clinical, like when we're working with real patient data, we tend to see that the different covariates will impact different parameters within this model. And that helps us to stabilize our estimates for how these likely behave in a patient. So given even very early data, if we know that they're squamous histology or that it's a female, we can make a reasonable prediction for where the tumor trajectory would likely go. That's what I was planning to present. I think at this point, I would open it up to questions.
Great, thanks, Jacqueline, for that presentation. We've got some questions, and so I'm gonna start with this one. So you showed the tumor trajectory model. This is what I believe is known in the literature as the generalized Stein-Foho model. Mm-hmm. And so the question is, how flexible is the tumor growth model? What sorts of shapes can it capture, and what sorts of shapes it may not be able to capture? for example, sometimes we see in immuno-oncology the initial growth followed by the shrinkage and so on. So can you just comment a bit about that particular model and how and if it can be expanded?
Sure. So let me show this slide that I didn't include, but it does break down the model into some of its pieces. And then I'll get to what are the shapes that it can't accommodate. So I'm showing here the relative change, the percent from baseline in blue for a given patient. And in green is the response, the sort of treatment response. This is computed from F and the growth rate, the shrinkage rate, KS. In gray is the portion of the tumor that is not responsive to treatment. So even though this patient has a fairly rapid, decay among treatment responsive cells, because over, close to 80%, over 80% of the cells or of the tumor diameter is not responding to treatment, we see a relatively modest shrinkage. KG in red is showing the growth of the treatment resistant cells. And so this is growing pretty slowly, and that's why we see this very shallow decay. What this model does not allow for is like a delayed response to the drug, where we sometimes see the tumor will grow before shrinking, or we'll see that there might be a lot of growth before shrinking. We can also see multiple sort of bumps in the tumor trajectory if a patient changed treatments or other features came into the tumor size. I think in the context of immunotherapy, the most common one that we're not accommodating is the delayed onset of treatment. When we fit this model we have a version of this model that includes a delay term that allows treatment onset to be later. But another way that we can fit it is to allow F to be a time varying parameter per patient. In that case, it also allows us to accommodate this. This is the type of thing that we tend to adjust depending on the trial data that we see. So in a lot of the data that we've been working with, the delay term, when it's estimated, is not impacting survival. And very few patients that we've seen have had the delay term. But that may be something that is drug specific or cohort specific, and we just may not have seen data for that cohort yet.
Okay, thank you. Thank you for that. Another question is, I think you were showing some simulations. A lot of this was simulations, but then there were a few plots for the distributions from real data. So I assume you have tried this with real data. What are some of the challenges there, and particularly how do you deal with censoring of tumor measurements?
Yeah, so the challenges with real data are twofold. One is that not every patient's trajectory fits the model equally well, right? So we do have sort of heteroscedasticity in the noise term on this SLD model, on the tumor size model. But in addition, we see that in real data, cohorts do not always line up, right? So we might have a cohort with PD-L1 expression greater than 25% and another cohort with PD-L1 expression between 50 and 90%. And so these cohorts sort of loosely overlap, but there's, in order to analyze the data well for data that is sort of partially discretized or discretized inconsistently, there's a layer of complication that we want to add to the model to accommodate those types of covariates. The second challenge with analyzing real data is that we want to investigate which covariates enter into which parts of the model. The tumor size model itself has some non-identifiabilities. So we need to stabilize these estimates for the parameters by including data for a lot of patients. And that often requires just many iterations of trying different covariates and exploring the data. The second question around censored tumor size measurements is sort of a challenge for two reasons. The first is that the tumor size measurements are not censored completely at random. So patients who have a metastatic event or who progress on treatment for reasons that are not manifested in the tumor size data, their data gets censored early. And similarly, patients who, and those patients have worse outcomes than patients whose tumor trajectories are continued to be measured. And in the clinical trial context, patients will drop out of the tumor size data collection when their disease progresses, and then they switch over to a new treatment. So we do a lot of testing on the tumor size model to ensure that there's no bias in the estimates when we truncate the data early. We also did a lot of testing on the calibration of that model using simulation-based calibration. For simulation-based calibration, we're simulating data from the prior and then fitting the model and testing that we're able to recover the prior distribution from the posteriors over many iterations of simulated data. And then we repeat this process where we subsample from we sort of simulate data for the observed covariates in the clinical trials. And there we do see some, there we want to test to make sure that we're able to recover the parameters that are used to simulate the data.
Okay. So the next, I think I can try to answer it, and maybe Jacqueline will have something to add. And it's a question of how did you defend your choice of building a Bayesian model versus a frequentist model to your supervisor or other stakeholders? So, we're working on this. We're lucky not to have supervisors, but we do have stakeholders. And so generally speaking, those people that work with us don't have an objection. So if a particular stakeholder has principal objection to Bayesian methods, it's unlikely that we're going to be collaborating with them. So sort of by design, we tend to work with people who are open to it. And more specifically, though, maybe a sort of an answer to a regulatory angle. As you, I'm sure know, the late stage trials that have to be analyzed with frequentist methods, but that's not where we generally work, right? So in phase 1/2, the decision to pursue one or the other treatment forward is an internal decision by the trial sponsor, right? And so in that role, they're allowed to use any methods they want, and they want to sort of use the best methods they can and often make decisions with the limited information. This is where Bayesian methods are really superior, we think, and that's kind of how where we fit into the picture. But I don't know if you have any comments on that, Jacqueline.
Yeah, I think so there are two comments. One is that this is a very important decision that's being made, and we want to have a reliable estimate for how much knowledge we actually have, how much uncertainty is there. One of the challenges here is that the sources of uncertainty sort of compound, right? There's uncertainty in how we estimate the parameters of the tumor size model, and then there's also uncertainty in how that translates to survival. And we know from some of the analysis of our clinical cohorts that estimating that well and sort of in what conditions does it vary across different treatments is very important to getting reliable survival estimates, that not all tumor reductions are equal. And so there's that, we want to translate this uncertainty, sort of include all the different sources of uncertainty in our model in order to have a well-calibrated decision and enough information to make a decision. The second reason is that using a Bayesian model allows us to use all the data that we have, to use all the information. And so we're able to include data from the background studies, But we're also able to adjust the priors based on expert knowledge about the treatment and which treatments it's likely similar to and how we would expect it to behave in certain patients differently than others. And so having a mechanism, a principled mechanism for incorporating that kind of expert knowledge is very important when trying to make a decision with limited information, which is the situation that we're in now.
Great, thanks. So next question is, are there opportunities to leverage additional information from related but different active therapies from the same drug class, e.g. borrowing information from patients and existing immune checkpoint inhibitors to stabilize estimation for new immune checkpoint inhibitor?
There absolutely is an opportunity to do that. That's something that we are testing in both simulated data and in real data scenarios. One of the areas where we can see that operate most effectively is in the TKI and in the chemo treatment classes. Right now, most of the immunotherapies, each pharma company has one of each type of immunotherapy. And so we don't know to what extent similar immunotherapies behave similarly in patients. And so we do want to cautiously proceed down this path, but we are seeing that the different treatment classes may behave similarly within a treatment class.
Okay. Let me ask, what do you think are some of the more promising model expansion opportunities, right? This current model, if I understand this right, built for the on-target lesions only, right? And it's a sum, based on the RECIST measurement of sum of longest diameter observations. Some of the information gets compressed, right, from that and from excluding other things. So what do you think? What do you think are some of the model expansion opportunities?
So the first category of model expansion is in the extracting the most signal from the tumor size data or from the biomarkers we have access to. That would include things like splitting the on-target lesion diameters by location and summarizing, lesions that are in the liver separately from lesions that are in the brain, separately from lesions that are in the lung. And one of the features of the way that tumor size is measured now is it's designed to be sort of to provide one sort of summary of the biomarker itself. So it's a simplified value in order to aid interpretation. By building out a model such as this one, it allows us to extract more signal from that early data and include more biomarkers and integrate them into a survival prediction in a principled way so that all of these sources of information can be included in the decision in a rational way. So now we can start to split out the lesion by location, but we can also include additional biomarkers such as circulating tumor DNA or the expansion of T cells in response to an immunotherapy. And ideally, we want to capture biomarkers that reflect the mechanisms of action for the drugs under consideration that are being tested.
Cool. Okay. Very good. What about multiple biomarkers, right? So there's lots of work is going on in trying to identify other markers. ctDNA comes to mind. There are lots of others that will be coming out. In terms of this model structure, can it be used to assess the efficacy of those markers? Can we include, multiple markers that, and what can be done from that perspective?
Yeah, so we can include multiple markers. It is a very natural extension of the model to include multiple markers. And in fact, right here, even though we're using tumor size, we're actually extracting sort of two signals from that tumor size data, and we can include additional markers in an analogous fashion. The second thing that we can use this model to do is to identify cases where the tumor size is more or less useful in predicting survival, or to what extent the tumor size is actually mediating the drug effect. We are seeing cases where, you can imagine a case where the drug effect is completely mediated by tumor size. The drug, say in a growth inhibitor type therapy, where the goal is to shrink the tumor and the patient is going to get better to the extent that the tumor shrinks. But a therapy that sort of minimizes the metastasis risk may not be manifested in tumor size at all. By looking at the tumor size and the survival data for these types of therapies, we can identify gaps where our longitudinal biomarkers are not capturing that treatment effect adequately. So that's an area of that would tell us which biomarkers to prioritize in evaluating them.
Okay, well, thank you very much. I think we'll stop here a little early. Thanks for this presentation, Jacqueline. What we're going to do is we're going to stop broadcasting now on the main stage. Still have a little bit of time. If you want to hang out in the sessions, I think you can still use networking, hang out there, or if you want to talk to Dan Lee and ask him some Stan questions, that's going to start at 1 p.m. Eastern, so about 8 minutes from now in the sessions. Otherwise, thanks a lot, everyone, for joining us. We have another cool event planned for next month. We're going to be talking about communicating uncertainty. Then we'll let you know shortly when that's going to be out. All right, so thanks, Jacqueline, and thanks, everybody, and we'll see you soon.